Hello everyone, welcome back to our Plante. In today's lecture, we'll learn all about the life cycle of angiosperms, part 5, in a simple and easy to understand way. So, let's begin. The life cycle of angiosperms, part 5. Second polar nucleus also unite with them. In Zea and Fritillaria, the polar nuclei with them. In several cases, the endosperm nucleus starts its division even before the first of the oospore. Therefore, the endosperm is at an advanced stage of development when embryo development starts. On the basis of method of formation of early stages of endosperms, the endosperm can be divided into three types. 1. Nucleotype. In this type of endosperm, the nuclear divisions are not accompanied by wall formation. The number of nuclei produced may be few, 4 to 16, to many, several hundred, depending on the size of the embryo sac. The nuclei are pushed towards periphery due to formation of a large central vacuole. The wall formation generally progress from the periphery towards the center, or from apex towards the base. For example, Fritillaria, nuclear type of endosperm. 2. Cellular type. In this type of endosperm, the first and the following divisions of the nucleus are accompanied by wall formation. In this way, the embryo sac is divided into many chambers. The first wall is usually transverse. 3. Helobial type. In this type of endosperms, the first nuclear division is followed by a transverse wall, resulting in the formation of a chalazal and a micropila chambers. In chalazal chamber, the nucleus divides and the division is followed by wall formation. In micropila chamber, the nucleus divides by free nuclear division, i.e., the division of the nucleus is not accompanied by wall formation. This type of endosperm is called helobial, as it occurs frequently in the helobials. Since the endosperm is meant for storage of food, its cells are usually thin-walled, large, and isodiametric. The stored food materials are starch, oils, fats, proteins, etc. Cellular type of endosperm. In members of the family Gramineae, the outermost layer of the endosperm act as a cambium and produce a series of thin-walled cells towards inner side. These cells become filled with starch and cease to function after some time, and are converted into alerone layer. It is considered that this layer secretes an enzyme which assimilate the food stored in the endosperm and makes it available to the embryo. In many seeds, no endosperm is seen at maturity, although it is formed at initial stages of embryo development. In these cases, during development of embryo, the food stored in endosperm is utilized by the developing embryo and endosperm is completely exhausted. Such seeds are called exalbuminous or non-endospermic. Example: gram, pea, bean, etc. In these seeds, the food is stored in cotyledons. In other cases, the endosperm is not completely exhausted by the developing embryo. Such seeds are called albuminous or endospermic. Example: in castor oil seeds, grasses. In many cases, the endosperm develops and completely fills up the nucellus space, and no nucellus is found in the seed. However, in some cases, as in Nymphaea mirabilis jalapa, the nucellus persists and develop into a nutritive tissue called perisperm. This is present in the seeds usually in the form of an apical cap. Rare cases, parthenogenetic development of the endosperm, i.e., before fertilization, is also reported. Ruminate endosperm. In some families like Palmae, Rubiaceae, Ananaceae, the endosperm is irregularly ridged and furrowed. This type of endosperm is termed as ruminate endosperm. The ruminations of the endosperm develop as a result of invaginations of the outer tissue, which penetrates deeply and appears as dark wavy bands in the Mautra seeds, or sometimes it develops as a result of an unequal elongation of the cells of any one layer, or the only layer of the seed coat as in Passiflora. Mosaic endosperm. In some angiosperms, there is lack of uniformity in the tissues of the endosperm. Such endosperm is termed as mosaic endosperm. Example, in Zea maize, the patches of two different colors are seen, white and yellow, forming a sort of irregular mosaic pattern. Weber, 1900, was of the view that the second male nucleus fails to fuse with the polar nuclei, and both divide separately, giving rise to nuclei of two distinct characters. These nuclei intermingled during free nuclear divisions, resulting in the development of mosaic endosperm. Clark and Copeland, 1940, provided most reasonable explanation for the development of the mosaic endosperm. According to them, there is either aberrant behavior of the chromosomes in mitosis, or there are somatic mutations which explain the problem. Nature of the endosperm. In gymnosperms, the endosperm is a gametophytic tissue, haploid, as it develops directly by the continued free nuclear divisions of the functional megaspore, while in angiosperms, the endosperm develops from the primary endosperm nucleus, which is normally formed by the union of two polar nuclei and a male nucleus, thus giving rise to a triploid tissue. The nature of endosperm has been a subject of controversy and discussion. Hofmeister, 1858, considered the endosperm of angiosperms as a gametophyte tissue, just like those of gymnosperms, with one difference, that its development remains checked till the entry of pollen tube in the embryo sac. Fusion of polar nuclei is also an act of fertilization. Therefore, the endosperm is a diploid tissue which serve as a storage tissue for the true embryo. Nawashins, 1898, discovered double fertilization and triple fusion. Sargent, 1900, supported Nawashin's discovery, and this discovery changed the views regarding the morphological nature of the endosperm. Strasburger, 1900, opposed Nawashin's and Sargent's view and suggested that triple fusion was not true fertilization, but a vegetative fertilization, which stimulates growth only. He suggested that endosperm is a belated vegetative tissue of the female gametophyte, stimulated in some way to develop by nuclear division, which provide food to the developing embryo, and the female gametophyte in angiosperms is much reduced and has no reserve food. This means that the development of endosperm in angiosperms takes place in same manner as in gymnosperms. But in gymnosperms, the endosperm is haploid, while in angiosperms it is generally triploid. Therefore, the view is not acceptable. The fusion of second male gamete with the polar nuclei serve two functions. 1. It stimulates the development of endosperm. 2. It helps in the formation of a tissue, which is physiologically more suitable for the nourishment of the embryo. Thus, it can be concluded that the endosperm in angiosperms is neither diploid nor haploid, but it is a undifferentiated tissue, which shows different degrees of polyploidy, from 2n to 15n. For example, when Oenothera, 2n, in Polygonomus, and most of the angiosperms, 3n, on Ditopelanthus, 4n, in Fritillaria, 5n, in Peperomia, 9n, in Peroromia hispidula, 15n. The direct effect of pollen on the characters of the tissues surrounding the embryo, endosperm, is termed as xenia. The term was introduced by Fouquet in the year 1881, but now it is used to indicate the effect of pollen in endosperm. For example, in Zea mays, white and yellow endosperms are present in two different races. If the pollens of yellow endospermic plant are placed on the stigma of white endospermic plant, the hybrid produced as a result will have yellow endosperm. The endosperm nucleus is formed by the union of two polar nuclei, Y and Y, and a male gamete, Y. Thus the endosperm contains three sets of chromosomes, Y, 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 and since it possesses a dominant factor for yellow color, therefore, it develops an endosperm of yellow color, although the ovule belongs to the white parent. Metaxonia. The effect of pollen on the seed coat, or pericarp, lying outside the embryo sac, is termed as metaxonia. Swingle, in the year 1928, found that in Phoenix dactylifera, at the time of maturity of fruits, Their size can be made to vary according to the type of pollen used in fertilization. According to him, the embryo and endosperm secrete hormones, which diffuse out in the pericarp and seed coat and exert a specific influence on them, resulting in certain specific variations. Significance of double fertilization. In angiosperms, the megaspore starts germination in the same manner as in gymnosperms, but the nucleus of the megaspore divides to produce a large number of nuclei by free nuclear division in gymnosperms, while in angiosperms, the nuclear division stops abruptly at 8 nucleate stage. During fertilization, one of the male gamete unites with the egg, and the other male gamete unites with the fusion nucleus to form endosperm nucleus. The endosperm nucleus rests for some time, and then starts free nuclear division, resulting in the formation of endosperm tissue. Therefore, it can be safely concluded that fusion of second male gamete with the fusion nucleus during double fertilization provides a stimulus without which the fusion nucleus would have remained inactive, and the process of free nuclear division, which had been terminated at 8 nucleate stage before fertilization, remains suspended for good, resulting in the non-formation of endosperm for the developing embryo. The process of double fertilization serves as a safeguard against unnecessary waste of energy by the angiospermous plants. Unlike gymnosperms, the endosperm formation in angiosperms follows fertilization, and in case the fertilization fails for certain reasons, there is no endosperm formation. On the other hand, in gymnosperms, the endosperm formation is not linked with fertilization, resulting in the wastage of energy, because the endosperm is there and the embryo is not. Xenia and metaxenia are also brought about through the courtesy of double fertilization, which leads to endosperm formation. Apomixis, when sexual reproduction in a flower is replaced by some form of asexual reproduction. The phenomenon is called apomixis. During this phenomenon, structures concerned in sexual reproduction are involved. It may occur by various forms, such as 1. Parthenogenesis. In this case, the embryo develops from an unfertilized egg, i.e., there is no fusion of male gamete and the egg cell. It is a rare phenomenon in angiosperms and is found in Phalictrum, Erigeron, etc. In these plants, the meiosis does not take place during megasporogenesis and the egg nucleus is diploid. In some plants, the embryo develop from a cell of embryo sac other than the egg. 2. Apogamy. Sometimes the embryo develops from a cell of embryo sac other than the egg. This is known as apogamy. This phenomenon is noted in Alchemilla, Allium, Iris, etc. The embryo developing from a synegid or antipodal cell may be haploid or diploid. Sporophytic budding. Sometimes the embryos arise by budding from the cells of nucellus or integuments, diploid cells of sporophyte, which project into the embryo sac. This is known as sporophytic budding, for example, in citrus, etc. If you have any question, then ask in comment section. Thanks for watching and see you in the next class. Polyembryony. In many angiosperms, more than one viable embryos develop in the seeds. Formation of more than one embryos in an ovule is called polyembryony. It is rare in angiosperms and more common in gymnosperms. The polyembryony arises in various ways. 1. The oospore produces more than one embryos during development as a result of splitting. This is termed as cleavage polyembryony and is found in allium, citrus, etc. Additional embryos called adventitious embryos may be produced as a result of sporophytic budding, e.g., in lemon, from cells of nucellus, and onion, from cells of integuments. An ovule may contain more than one functional megaspores, which develop into embryo sacs and oospheres, which on fertilization give rise to more than one embryos. Embryos may be formed from synegids, in addition to the embryo formed from ovosphere or from antipodal cells. Seed formation. The seed develops from the ovule after a few changes. The two integuments develop into two seed coats, the outer testa and the inner tegmen. In some seeds, the testa and tegmen fuse to form a single coat. The micropyle of the ovule persists in the seed in the form of pore, while a small scar, the hilum, represent the point of attachment of the ovule to the funicle, and it can be seen on one side of the seed. In some seeds, an additional investment grows up around the testa and surrounds the seed. It may develop from placenta, funicle, or micropyle, and is known as aril, as in lychee. In certain seeds, a small fleshy outgrowth of the seed is formed at the micropyle called caruncle. Example, in castor oil seed. Nucellus is generally exhausted. In some cases, it persists as a small thin layer called perisperm. The synergids and the antipodal cells are completely disorganized. Development of fruit. The stimulus of fertilization result in the formation of fruit as a result of changes in the ovary walls. The style and stigma dry up and disappear, and the ovary enlarge. The wall of ripened ovary form the fruit wall or pericarp, which may remain soft and fleshy or become dry and hard. When the ovary of the flower grows into the fruit, it is known as true fruit. But sometimes other parts of the flowers, such as receptacle or perianth leaves, take part in the formation of fruit. Such fruit is called false fruit or pseudocarp. Example, apple, pear, mulberry, pineapple, etc. Parthenocarpy. As a rule, the development of ovule into seed and ovary into a fruit takes place after pollination and fertilization. In most species of flowering plants, the failure of pollination results of seed and fruit formation. But in some plants, the fruits are formed even in the absence of pollination and fertilization. Such seeds are seedless and are said to be parthenocarpic, while the phenomenon is called parthenocarpy. The examples are navel oranges, grapes, bananas, pineapples. Megasporogenesis, megagametogenesis. Type negaspore, mother cell division. I division, 2, division, 3, division, 4, division, 5. Mature embryo sac, monosporic, 8 nucleate. Polygonum type, monosporic, 4 nucleate. Oenothera type, bisporic, 8 nucleate. Allium type, tetrasporic, 16 nucleate. Peperomia type, tetrasporic, 16 nucleate. Pennea type, tetrasporic, 16 nucleate. Drusa type, tetrasporic, 8 nucleate. Fritillaria type, tetrasporic, 4 nucleate. Plumbagella type, tetrasporic, 8 nucleate. Plumbago type, tetrasporic, 8 nucleate. Adoxa type. Let me know if you need help with anything else. If you found this video helpful, please give it a thumbs up, share it with your friends, and don't forget to subscribe for more easy biology lectures. Give answer the following questions. 1. What is sporophyte? 2. What is gametophyte? 3. What is endosperm? If you know the answer of the following question, then write in comment section.